is Karina, and this is a video presentation on the basic principles of genetics and its applications to HBV. So I'm going to start off with the basic building blocks of DNA, which are the nucleotides. So right now we're looking at cytosine, and you can see there's a phosphate group, a deoxyribose sugar, and a base. Differentiate the nucleic acids based off the bases, and cytosine is an example of a pyrimidine because it only has one ring in its base. And then you have the purines, which consist of two rings fused together instead of one, such as adenine. So my weird way of remembering purines is I think of adenosine as the apple and guanine as the grapes, and therefore they're pure sugar, and therefore they're the purines, because the purines are the ones with the bulkier groups, the two fused rings that are attached to that deoxyribose sugar. And then I like to think of the pyrimidines as the ones without as much sugar. So we got thiamine as the tomato and cytosine as the carrot, and there are sad and not sweet. So apple and grapes, pure sugar, and tomatoes and carrots, sad. And each base comes with a pair. So I remember as at GC, so adenosine and thiamine go together, and guanine and cytosine go together. These bases are paired together through hydrogen bonds, and I made guanine and cytosine extra cozy because they have an extra hydrogen bond compared to the other pair. So my way of remembering things is kind of weird, so if you have a better one, just ignore everything I just said. So now I'm going to go over DNA synthesis, and with transcription, you have to remember that it goes from 5' to 3'. So the 5' carbon is the one that has the phosphate group attached to it. So the OH groups on those carbons are going to interact with each other, and a water molecule is going to leave, and what you have left is a phosphodiester bond. So here, DNA polymerase is reading the DNA strand 3' to 5', but synthesizing 5' to 3'. What makes HBV unique is that it has a partially double-stranded circular genome with a DNA polymerase attached to it. It also has the smallest genome of any known human virus. Here it is next to a penny for reference. Once HBV enters the circulation, it eventually attaches to a hepatocyte. Once inside the cytosol, the virus uncoats and enters the nucleus. Here, DNA polymerase completes the synthesis of the double-stranded DNA ring. The DNA is then used as a template for the host RNA polymerase II. The mRNA then travels into the cytoplasm to be translated by a ribosome. With the ribosome, there's also tRNAs that have specific amino acids that match up to the specific 3 bases. Translation is initiated with a start codon by the ribosome, and the start codon that's recognized for this virus is going to be one that codes for methionine. Several mRNAs are then translated into various proteins. HBC, for example, is one of the core proteins that surrounds the DNA. The viral mRNA that codes for POL, which is a reverse transcriptase, is packaged with a POL protein into a viral capsid. POL then reverse transcribes the mRNA into the circular DNA along with the partial double strand. This viral-laden core then enters the ER and buds off while picking up the envelope proteins such as HBAG and HBsAg. So this is the completed virus with the envelope. It then goes out to the cell membrane and leaves to infect another cell.